What is an antibody? Antibodies are proteins that are manufactured by your B plasma cells. And those B lymphocytes that are doing this particular job will create proteins that will, well, diagrammatically, we could represent them along these lines here. So this would be the light subchain, which is combined with a hinge-like part of a protein onto a heavy subunit here. So these areas that we're drawing now are, for example, the non-variable regions. They are constants. In other words, all antibodies will have this particular structure. You notice it's a Y structure. It looks like a Y. Now these arms here, these light arms, as opposed to the heavy subunit that's created down the base here, these light arms have a variable region on the end. I've drawn two different sorts here, and you can see that they would fit different structures of your antigens or antigenic surface structures there. So this part here is your variable region. So antibodies are manufactured to be able to cope with different types of antigens that are present in the environment. So let's think about what an antigen is again. For the purposes of the immune system, an antigen is something that can create an immune response. Not all antigens will do that. Remember, you've got your self-antigens. But your antibodies will function by trying to trap the antigenic structures, those epitopes of the antigens, so that it can make them ineffective. There are four main ways that this can happen, and this appears in your textbook too. One of the ways that antibodies are effective is that they cause agglutination. An agglutination means to stick something together, and it creates a mesh or a network. So if you could imagine these little Y-shaped antibodies, if I drew them here, and you've got, here are the antigens that they're responding to. So here are your little Ys, they will accumulate lots of those, and this whole bit of agglutination means you create a mesh or a network of them. They stick together, they're not capable of moving along further. Another way that they function is called opsonization. Now, opsonization you're familiar with from our last chapter of work, and opsonization is when you're going to use these to tag particular pathogens for their removal by phagocytosis. So in tagging them, if it was on a surface, for example here, here's the epitopes on the antigen here, and if we had the antibodies attaching to those, they're acting as markers that will attract your phagocytes to that area. Another way that they work is neutralization. Neutralization, if antibodies are liberated into the bloodstream, wherever they're present with an antigen, it means that, well, firstly, your antigen on a particular surface of a cell, it's trying to get into this cell over here. So it's got to have particular binding sites that it recognizes and would be able to dock with in order to get into this otherwise healthy cell. So if the antigen has this particular shape, then the antibody surface shapes might look like this. And what the neutralization component is it's taking care of those active binding sites so that now they can no longer go into their target cells. So that's neutralization. And the last way that they're going to manage is called complement activation. In complement activation, you're doing exactly that. You're going to create the cascade of complement proteins, which will increase what they're doing and create a high level of an immune response based on all those proteins that are present within your blood system.